Hello, and I'm going to do the same thing with 7b as I did with A. I'm going to cut it into two places because of the length. And what we're going to do first is we're going to talk about DNA and RNA, the differences between it. Then we're going to talk about transcription, how we actually change DNA into RNA, and then translation, how we take the RNA and turn it into proteins, which is everybody. If you take a look at this picture, if you remember, we have a 5'3" strand and we have a 3'5" strand. And it's important to remember this because when we actually do the transcription, turning DNA to RNA, we're going to use the 3'5" strand because we really want the RNA to be a 5'3" strand. And remember, actually the full name for DNA is a deoxyribonucleic acid. Parts of the nucleotide, we still have a sugar and phosphate, which makes up the backbone, and we have the nitrogenous base, which makes up the rest of the nucleotide and makes up the rungs of the ladder. And then there's double helixed, this strand and this strand double helix with each other. Numbers of strands, we have two. We have the 5'3", the normal one, and we have the backward one, which is the 3'5". And again, that has to do with the position of the 3 and 5 sugar. If you have a 5 sugar at the top and then a 3, or on this side we would have the 3 at the top because it's close to phosphate, and the 5 on this side. So we have a 5'3", and we have a 3'5". Name of the nitrogenous bases. Remember, there's the leading, there's the lagging, and remember we have adenine, thymine, cytosine, and guanine, and the base pair adenine and thymine, and cytosine and guanine, or guanine and cytosine. And the function of these, this is the boss. This is the section that never leaves the nucleus. I guess during mitosis, the nuclear membrane breaks down, so it sort of does, but in terms of the transcription and translation, it never needs. And it's got the instructions to build the proteins. In other words, it's got the genes. RNA, full name of it, is ribonucleic acid. Parts of the nucleotide, we still have a sugar phosphate backbone, and we have the nitrogenous base, except that we only have one strand of this, so we don't have the rungs. At least we have like half a ladder. Names of them, we have adenine and cytosine and guanine, but instead of thymine, we have uracil. And the A's and the U's stick together, and the C's and the G's, just like always, stick together. Remember that anytime we have an adenine on the DNA, we're going to get a uracil on the RNA. And the function of this one, this is going to assemble or put together the amino acids, which are going to make all the proteins. And remember, your body is the protein. This does all the work. DNA has the code, RNA does everything else. So DNA, we actually have the phosphate, we have the sugar, and we have the base. In RNA, we have the phosphate, we have a sugar, and we have a base, except this is a deoxyribo. In other words, the oxygen is taken out of this hydrogen, and there it is. That's a hydroxyl, and that's what actually the deoxy is the DNA, and the oxy is the RNA. Remember, we have T's and A's and C's and G's, and here we actually have U's and A's and C's and G's. Double-stranded 5'3", 3'5", and a single-stranded 5'3". If you take a look at it, there's the hydroxyl and there's the hydrogen, and we have the hydroxyl, it's a ribonucleic acid, and we get rid of the oxygen on it, it's deoxyribonucleic acid. RNA actually comes in three different types. We have mRNA, that stands for messenger. This is the stuff that's actually made inside the nucleus. It codes for the DNA. We have 3 nucleotides that actually make codon, and then 3 we make a codon, and then the next 3 we make a codon, and thus far. And then made in the nucleus, it carries the code to the ribosome. The DNA and the RNA, if you have TAC, you're going to end up with AUG. Remember, the T's go to A's, but A's now go to U's, and C's go to G's. And then G's goes to C, G goes to C, and A goes to U. Remember, an RNA has uracil. And then we actually do have what are called exons and introns, and the exons actually in the messenger RNA come down, the intron comes down, codes out, but by the time we make a mature messenger RNA, we cut out the introns, and the introns are basically things that don't code for proteins. So we get a 5-strand and a 3-strand, and we actually break away the 3-strand, and the 3-strand actually makes another 5-strand, just like it would in regular DNA synthesis, except that this 3-strand is the only one that's red and creates a 5-3-strand of messenger RNA. And these are actually the codons, and the codons actually transfer into different proteins. So methionine and different ones actually code for different other ones, and we'll talk about that in a few minutes. The next type is called tRNA, and this is for transfer. This actually makes a key-shaped or a T-shaped material. The anticodon stands down here. This will actually have a CGI type of format, and this one actually up here is where we actually code for the amino acid. This is where the amino acid that this transfer RNA carries from collecting it out in the cytoplasm and taking it down to the ribosome where we're going to make the protein. There are 20 common amino acids that make up all the proteins in a human body. We have ones that are called essential. That's these dotted lines that you see. They come in different groups, but these are the essential ones. These are the ones that we have to make sure we get from the food we eat because we can't get them from any other source. The last type of RNA, the last one of the three, is called rRNA, and this is the actual ribosome, and ribosomes are made of RNA. Remember, these things are made in the nucleolus of the nucleus, which we learned about in Unit 3. This makes up the ribosome, and this is where the proteins are made. Remember, proteins are these really coiled, ribbony kind of amino acid chains that actually help for creating things, as well as you have types that speed up things, slow down things, but basically it's for basically all your bodily products. Okay, so this is the messenger RNA. It actually is made up of two parts. We actually have the rRNA, we have the messenger RNA, we have the tRNA, and those are the three types of RNA, and the tRNA actually brings in the amino acids, which will actually come in and bond together to make the proteins. And they'll be peptide bonded, if you remember back in Unit 2. So, transcription. Transcription is actually where we take the DNA and we change it into RNA. And the way that works is inside the nucleus we have this chromatin. The chromatin actually gets a protein that comes through and unzips it. Remember, we have the 3 to 5 section, so we actually put the codon for messenger RNA. A's become U's, T's become A's, G's become C's, and C's become G's, and these actually code for different things. So this is going to be the messenger RNA, and you can see that they're actually pulling in, they're actually trying to line up with the 3-5 band of that DNA. So transcription, this is happens in the nucleus. This is where we copy the DNA and actually code for the messenger RNA. Leaves the nucleus. So here's the DNA. It actually coded for the 3 C's become G's, A's become T's. So that's the 3-5 strand. This is the messenger RNA. This is the gene. The gene's made up of a whole bunch of different codons, and the messenger RNA will then leave the nucleus. There's where it leaves the nucleus. It'll find a messenger RNA. The messenger RNA will find the rRNA, the ribosome, and then what will happen is the T will bring in the tRNA. Will actually bring in. They'll code for the codon. Will be code for the anticodon, and the anticodon will bring in the amino acid. The amino acid will do a process called elongation. It'll actually get to the right format until we finally get a stop codon, which we'll talk about later on. The stop codon will end the protein, and then the messenger RNA will move over and find another ribosome to make more protein until the DNA decides that that protein has been made enough, and then the messenger RNA is lysed. The ribosome will go on and actually maybe latch onto the same messenger RNA, the same type or a different type. Messenger RNA actually codes for the protein. The rRNA is going to actually help make whatever protein the messenger RNA is supposed to make. And the tRNAs will come in at a very set path to make sure that the anticodon on the tRNA is the same as the codon on the mRNA. So translation happens at the ribosome. It's where we build the amino acid sequence, which is actually provided by the messenger RNA code. So here comes the messenger RNA. We had GUA, we have CGG, CCG, and we have GAC. We have three groups of nucleotides which actually make up the codon. There's the next codon, there's the first codon, and it actually is getting read. Actually, I can't tell which way it's getting read because we actually need to see the three sites that are in here to determine that. Looks like it's being read this direction. tRNA group comes in. There are three tRNA nucleotides that make up the anticodon. That's the CUG, and that comes in and actually codes for the messenger RNA, and that'll bring in a very specific amino acid. And then this one brings it in and will make a very specific protein. This is the codon wheel, and the way the codon wheel actually works, if you have AUG, first you read on the inside, and you actually see you have the four different types there. So you can start with a U, you can start with a G, a C, or an A. In this case, we're starting with A. And the next one we go to a U, and the next one we go to a G. And this actually is the codon for methionine. And this is the DNA, or the messenger RNA message that comes out. It is not the anticodon, it's the codon that gets read with a codon wheel. GUA, we go G, U, and A, valine. CCG, CCG, proline. And then GAC, GAC, aspartic acid. And then if we do the same sort of things, you see AUG, you start here, there's A, go to U, and then go to G, which is way the heck over here. You get methionine. I like the wheel much better than this data board, or the basically the column setup. And then we have UAG, and if we do UAG, you see that's the stop codon. Start codon is methionine, stop ones can actually be these three. So what happens is it starts reading at the methionine, or the AUG codon, starts reading there, pops in all these different amino acids, and it will keep popping these things in until we finally get to one of these three, and that actually tells the RNA to stop creating that protein, and that protein is now set. Okay, and that's where we're going to stop. Thank you for stopping by. Bye.